Okay, so today we are going to finish up talking about how our mean arterial pressure or MAP is regulated. Okay. So last time we were talking about the baroreceptor reflex. So we went through all the factors included here. What we were talking about, right, was we know we have an interrelationship between flow, pressure, and resistance. We know that our flow is really our cardiac output, so we can reformat this equation such that our mean arterial pressure, or pressure difference, is going to end up regulated by our cardiac output, which is our heart rate and our stroke volume, and our total peripheral resistance. Okay, so we were talking about factors that can raise our blood pressure back up when it drops, right? Which is the situation we're dealing with when we go through our baroreceptor reflex. Okay, so we're noticing that if we have a sudden drop in blood pressure, your body wants to reflexively lift that blood pressure back up so you don't pass out. Okay, so we've got control centers for this. Really, the way we're sending our effects out to actually have the effect of pulling our blood pressure back up is through upregulation of the sympathetic part of our nervous system and turning off any parasympathetic effects that we may have at rest. Okay, so when we have this increase in sympathetic activity, this is really external or extrinsic regulation of our mean arterial pressure. What ways we can do it? We can raise that heart rate up, right? So we can raise heart rate by increasing sympathetic activity and decreasing parasympathetic activity at the same time. We can raise our stroke volume, so how much blood your heart is pumping out per heartbeat. Okay? So we have a couple of ways that we can do this, but they are both linked back here to that sympathetic activity as well. Okay. One piece is that we literally have sympathetic nerves going to the ventricles of the heart, right, that cause them to contract more and more rapidly, right? So we increase ventricular contractility, squeezing more blood out, more stroke volume per heartbeat. The thing that we're tacking on that we hadn't talked about last semester, but now we have talked about in our study of veins, so the sympathetic nervous system also does affect the veins. Specifically, what it's doing is it's changing that venomotor tone. There's essentially tension in the wall of the veins. So when we make those vein walls more tense, increase their venomotor tone, what we are doing is we're decreasing their compliance. So we're making them behave a little more like an artery would, right, with the low compliance, meaning that we can build up pressure, right, when we have that same amount of volume. What causes the heart to fill, right, is a pressure difference between the veins coming into the heart and the blood left in the heart itself. So when we increase pressure in the veins by increasing that venomotor tone, what we're really doing is causing the heart to fill more. So this is measured by our end diastolic volume. We have a higher end diastolic volume, so more preload, more blood in the heart at the end of its resting phase, that diastolic phase. When we squeeze that blood out, we squeeze out more stroke volume. So our final way to lift that blood pressure back up is through resistance, right? So if we raise our total peripheral resistance, that also pulls our blood pressure back up, okay? So the way that works is mainly when we have our sympathetic nervous system giving out its norepinephrine, activating those alpha receptors in the smooth muscle of the arterioles. When we activate those alpha receptors, this causes vasoconstriction. Vasoconstriction, we have that radius of the arteriole go down, which means we're having higher resistance. So when we add up all that resistance to get our total peripheral resistance, we get a higher value. And all of this leads to pulling that blood pressure back up. Questions about this process? This is threading together many of the things that we've done in this unit. Kind of what we've been building to. Right, so we're going to add on a couple details today. Right. So for our long-term regulation, right, we don't want our nervous system to have to constantly be regulating this. So we've got the baroreceptor reflex that we just described, responsible for quickly changing blood pressure, just as like kind of compensation. But it's not actually changing whatever the problem is, right? So if you have a drop in blood pressure because you literally don't have enough fluid in your blood, you literally don't have enough blood, right? Squeezing those vessels a little harder using your nervous system is not going to fix whatever's going on with that, right? So we're not going to use the baroreceptor reflex to make a long-term change to whatever's going on. It's really more suited to things like, oh, you just stood up and gravity's kind of pulling on your blood. Situations like that, good for the baroreceptor reflex. But for long-term regulation, for long-term problems, we really want to use the kidneys in order to actually fix the amount of blood volume. But that's our renal regulation. Renal always refers to the kidneys. Okay. And that's going to be linked in with a bunch of hormones that help regulate kidney function and blood volume. So some of the hormones that are also involved in this process, this sort of longer term regulation. We've got our epinephrine, vasopressin, and angiotensin 2. We're not going to think about those extensively right here. But we have mentioned epinephrine before, so let's just like recap what that was doing various places, right? Okay, so our overall effect of epinephrine is going to be that it will increase our mean arterial pressure. Okay. So let's trace there what we know about our epinephrine. So our map is our heart rate, our stroke volume, and our resistance. Okay. So we talked a bit about epinephrine's effect on the arterioles when we were going through those arterioles. Right, we started out with the norepinephrine activating those alpha receptors throughout the body. And we were talking about epinephrine. This was the one where we had to think about concentration, right? So epinephrine is the one that is highly preferred by those beta-2 receptors, which are located only in the blood vessels going to your cardiac muscle and to your skeletal muscle. So under a low concentration of epinephrine, we had this vasodilation effect, specifically in blood to the heart and the skeletal muscle. Okay. That were the only thing that was going on. We kind of expect a drop in blood pressure, right? Dilation, less resistance, we'd expect a drop. Which is why it's important to know that as epinephrine concentrations rise, right, as we have a spillover epinephrine, as we have more epinephrine, which we will, epinephrine also binds to those alpha receptors out in the rest of the body. Right? So if we have enough epinephrine, if we have higher concentrations of epinephrine, it's going to end up doing the same thing that our norepinephrine did, right? Those alpha receptors prefer norepinephrine, but not so much that they won't take epinephrine if you've got a lot of it. So overall, the effect of a bunch of epinephrine is going to be to increase total peripheral resistance at the same time as it's shuttling what blood flow does exist to the heart and the skeletal muscle to work in kind of synergy with that sympathetic nervous system, fight or flight. response. Okay. So, because we're now remembering that epinephrine, if we've got enough of it, can bind to those alpha receptors, right? Those are also what we're responsible for our changes to venomotor tone, those increases in venomotor tone. Right, so we got alpha receptors in the smooth muscle, the veins, right? We have enough epinephrine. It's going to bind to those receptors in the smooth muscle veins. I'll clench up a little bit. That's our increase in venomotor tone, which we just walked through how that results in higher end diastolic volume, right? So more preload, more blood filling the heart at the end of its relaxation phase, and therefore greater stroke volume by the time we actually go through our whole cardiac cycle. Finally, epinephrine acting as a hormone does just like act on the receptors in the heart muscle itself. So when it does that, its effects are just like the sympathetic nerve effects. So we see an increase in heart rate, so faster heartbeat, as well as increases in stroke volume as it affects cardiac muscle cells in the ventricles. Okay. Vasopressin and angiotensin 2 are vasoconstrictors. Right, so really like every other factor that we have been discussing, they're going to increase your blood pressure, increase MAT. They do that mainly by modulating our resistance. So they're going to increase resistance on the whole when we see them. So we want to go back and think a little bit more about those receptors. So I just want to mention, right, we mentioned that we have low pressure baroreceptors before, but last time when we spoke, we were talking more about the pressure receptors, the arterial pressure receptors in the arch of the aorta and in those cardiac sinuses going up to your brain, right? So those ones were sensing pressure, right? Which is part of how we were getting a signal for this baroreceptor reflex, right? When we saw a drop in pressure using those arterial stretch receptors. So the low pressure baroreceptors kind of work in conjunction with those receptors in the arteries, but they're in different places. Okay. So we've got more receptors, this time in the walls of some of our big systemic veins, like the vena cava or something. As well as in the walls of the atria, so like in the heart itself, in the atrium, we have some of these volume receptors. Okay. So these are measuring not the change in pressure, right? So the ones in the arterial walls are checking how stretched out are those artery walls, how much pressure is forcing against the arteries. These volume receptors in the veins and in the heart itself are checking, okay, how much volume is there, okay? Which makes sense because our veins are where we have 60% of our blood volume at any given time, right? So that's really their job, is as this volume reservoir. Okay. So when we see a decrease in blood volume, this activates those receptors, and then this is going to respond in a way parallel to the baroreceptor reflex. So the baroreceptor reflex was sensing a drop in pressure, right? And we can pull pressure back up using all those different kind of fall down effects of the sympathetic nervous system. If we know that we have a decrease in blood volume, you don't have enough blood in your system, right? Like physically, you don't have enough blood. This is also going to go hand in hand, usually with the drop in pressure. It's just a different way to notice it, just a different way to sense that. So we're going to have synergistic effects as going along with those things that we talked about with the baroreceptor reflex. So there's not really going to be like a different kind of cascade of events for those volume receptors. They're just kind of going to go along with it. We do have some other kind of weird special things that affect regulation of our cardiovascular system and our mean arterial pressure. So we'll touch on a couple of those special concepts right now. Okay. The first one has to do with your breathing. So spoke briefly about breathing when we were talking about that respiratory pump as a way that we're getting venous blood into the thorax and then into the heart. Right? We saw that we have these changes in pressure in the thorax with breathing. A cool and somewhat mysterious thing is that breathing in and out actually does have a link to activity of your sympathetic and parasympathetic nervous system. So we'll see that sympathetic and parasympathetic nerves, when we get to our unit on the lungs, have effects on how you breathe and your breathing rate. But it's actually true that your physical breathing changes the amount of sympathetic activity and parasympathetic activity, kind of from the opposite side. So actually, when you inspire and you breathe in, this slightly lifts up the sympathetic activity going on in your body when you breathe in. And so it has a tendency to raise your heart rate when you are breathing in. When you breathe out, that's your expiration piece. This increases your parasympathetic activity a little bit. So you've got this nice back and forth as we breathe in and out. And this lowers your heart rate a little bit. So the speed of your heart and how it beats can change a little bit based on your breathing. So when you breathe out nice and long, you can actually kind of slow down your heartbeat, right? Slow down that kind of stress level overall. So as we see heart rate change, right? We wanted to look at the downflow effects of that just in that instant when you're breathing in versus breathing out. If our heart rate were up, we would expect our mean arterial pressure to be a little up. If our heart rate is down, we'd expect that mean arterial pressure to be a little bit down. But hopefully you're breathing in and out multiple times in a minute. So since mean arterial pressure is averaged over a minute, depending on exactly how you're breathing, you're not going to see real noticeable effects, except at that kind of instantaneous. Another reflex that we have, which we'll talk about more again when we talk about breathing in detail. But this reflex is going to be in response to blood gases. So this is your chemoreceptor reflex. So rather than sensing pressure, like those arterial baroreceptors, or sensing volume, like those volume receptors in your large veins and the atria of the heart, your chemoreceptors sense the chemical composition of your blood. So we're going to see that we have some kind of like up in our brain, basically. Okay. So what they mostly notice are carbon dioxide levels. So part of what we're going to see them doing is helping us like regulate the pH of the blood. We'll get to why carbon dioxide and pH are related. So backing up, these chemoreceptors noticing changes in CO2. And their main job is to regulate that amount of CO2 in the blood. They want you to not have a huge buildup of CO2. Okay. This means that they affect breathing, because breathing is going to be one of the ways that we can very obviously change how much of each of our different types of gas is allowed in our system. And they're also going to change our resistance a little bit. This is actually going to be a little different than when we saw our local effects of those gases on blood flow. Okay. So what happens when our chemoreceptors sense an increase in carbon dioxide, right? This is kind of a dangerous situation for your body, right? You're seeing change to pH, carbon dioxide buildup, bad, we kind of dealt with. But what your body does in response, at least from the perspective of our blood pressure, makes it seem a little counterintuitive. So we might think initially that we want to increase our blood flow, but what we actually do is we start by decreasing the heart rate. So this is part of the chemoreceptor reflex. What's going on there is that Your heart uses a lot of oxygen and produces a lot of carbon dioxide when it's beating quickly. So if you have a buildup of carbon dioxide, right, this means you may also be using up a lot of your oxygen, right? So if we slow the heart down in response to noticing this change, we can kind of try to dial that back, right? We have a slower beating muscle. It doesn't need as much energy, which means it doesn't produce as much carbon dioxide, and we're kind of preserving oxygen, right? We don't need as much oxygen if we're beating that heart a little bit more slowly. Okay, so that heart rate piece, we looked at just that bit, we would think that we'd see a decrease in mean arterial pressure in response here. But we don't actually want that, because we do want to maintain our blood pressure nice and even. We don't want to faint. We want to keep blood going to the brain, right? Even if we're changing stuff out in the body. So another thing that the chemoreceptor reflex specifically does is send signals out that are going to increase our total peripheral resistance in order to kind of counteract this. Okay. So this is a situation where we have two effects that kind of don't go in the same direction, right? So when we're looking at that mean arterial pressure, right, we know we can regulate heart rate, we know we can regulate stroke volume, and we know we can regulate total peripheral resistance. Okay. So what we see happening is that at the same time as it's decreasing that heart rate, to keep that blood pressure back up, it's also going to send out signals to conserve any resistance, to do a little bit of vasoconstriction, to pull that total peripheral resistance back up. And on the whole, that resistance effect wins out a little bit, actually. So we see a slight increase in mean arterial pressure in response to this chemoreceptor reflex, noticing that we have a buildup of carbon dioxide. Conversely, this may also be helpful when we think about the last time we talked about carbon dioxide and the blood, right? We were talking about active and reactive hyperemia, right? And how if we have that buildup of carbon dioxide in the tissue, right, we're essentially going to vasodilate at that local level and allow more blood flow to kind of wash it away. The other reason the chemoreceptor reflex is helpful, right, is that vasodilation from those local effects would be pulling your total peripheral resistance down. So it's important and helpful that this larger reflex coming with signals kind of from your brain is pulling that resistance back up, making sure that these other things that are going on in response to the carbon dioxide aren't making you pass out from lack of blood to your brain. Your temperature regulation is it's another homeostatically regulated variable, and it's actually more important to your body than your blood pressure is. So thermoregulation actually takes precedence over the baroreceptor reflex, right? And they are going to kind of have to fight each other a little bit, because the way that we adapt to a change in body temperature, right? If you're overheating, your body temperature is going up, right? In order to lower body temperature back down, I think about last time you had a fever, or last time you got really hot working out at the gym or something, right? One of the ways that we pull body temperature back down is through sweating. And when we're sweating, right, part of it, what we have to do is we want to bring lots of blood to the skin, right? So you have a lot of blood right there under the skin, so that as your sweat evaporates, that evaporated cooling also cools the blood that's now kind of in contact with your skin. So increases in body temperature create vasodilation to the skin so that you can lose heat to the environment and keep yourself cool, right? So that's what we want to do, like actively want to do, to avoid getting like heat stroke or something. So that's going to happen when you're overheating. But if we think about that from a blood pressure perspective, right, what we've just said for our blood pressure, yeah, order of our variables does not matter. What we've done is we've affected that resistance now, right? Okay. So we affected that resistance with that vasodilation, which is helpful for your body temperature because that's what's gonna let that blood cool as it's on the surface of your body getting cooled down. But overall, the effect this is gonna have, if you have vasodilation somewhere in the system, what we mean is we're lowering resistance. So we see here a drop in resistance causing a drop in mean arterial pressure. So this is part of why you can see like, or why you might feel that you get like kind of lightheaded or something when you're overheating a little bit. Like you might actually be getting lightheaded due to blood pressure effects, right? Because of this vasodilation and the fact that this thermoregulation takes precedence over our mechanisms for increasing blood pressure when it drops too low. Finally, we do remember that exercise changes the distribution of blood flow to our organs, as well as changing our total cardiac output. So overall, we're increasing a lot of blood flow to your skeletal muscle. We're increasing blood flow to the cardiac muscle, to the heart, at the same time as increasing our heart rate and stroke volume. We are also at the same time decreasing some of our total peripheral resistance, depending how hard we're exercising, depending exactly how much epinephrine is swimming around, right? But overall, these effects on heart rate and stroke volume hopefully lift that mean arterial pressure up a little bit. So we're going to break into the first bit of our next slide deck. So we're going to talk a little bit about the blood. So we're going to start by thinking about the components of blood. So we're going to be talking about the parts of the blood, and then we're going to dial in and think a fair amount about our erythrocytes, our red blood cells. Don't think we're going to get as far as a lot of clotting before the exam, but hopefully we'll manage to get through this first little thing. So before we begin, I want us to remember what our blood is, right? So we're describing the plasma, right? We've described like the plasma getting filtered when we're calculating our net filtration pressures. Okay, so what actually is the plasma? What's in there? Take another couple seconds. Let's take a look. Okay. All right. So we got a couple disagreements. So I am going to tell you that the plasma is mostly water. So when we refer to the plasma part of your blood, right, that was actually one of our forms of extracellular fluid way back in our first week of A&P 1, right? Plasma was extracellular fluid, as was our interstitial fluid, so fluid surrounding other types of cells in the tissue. The erythrocytes, the red blood cells, are the cells that plasma is surrounding, right? So the water itself and the things dissolved in that water are our plasma. So when we're talking about the net filtration pressure acting on fluid-free plasma, that's why we're just kind of thinking about it as water mainly, because water really is the main component of your plasma. Cool. So from composition standpoint, your average blood volume is somewhere around-ish 5 liters, 5.5 liters, okay? And we've got a slightly off-kilter split between your amount of plasma, which is about 55% of your whole blood volume. So we take a tube of blood and we let the parts separate out. A little more than half of it is going to be plasma. So that's going to be like water, electrolytes in there. A little less than half is going to be made up of your actual erythrocytes. So the red blood cells themselves. So when we measure the hematocrit, that's a word for talking about that relationship, that percentage. Basically, when we measure hematocrit, what we're talking about is how many red blood cells do we have, right? Like what percentage of our blood is made up of red blood cells. Okay. And typically it's somewhere around 45-ish%. There's a normal range that depends on a couple different factors. Okay. And then we have a tiny little bit of that test tube that would be filled with leukocytes, our white blood cells, as well as platelets, which are going to be one of the things that we're going to talk about as we begin talking about clotting, probably at the beginning of our next unit. So these are all things that we'd expect to see in the blood. So about 3 liters of that total of 5.5-ish liters is made up of plasma, so basically water. Okay. Then we've got another little less than 3 liters, so like just under 2, let's say, 2-ish. That's going to be the red blood cells. And then a teeny tiny bit, that's your white blood cells, your leukocytes, and your platelets. So what we're going to start thinking about are actually those erythrocytes, those red blood cells. So I'm just going to point out to you that all of our blood cells come from hematopoietic stem cells, right? So we start with one type of stem cell that leads to all of our different types of blood cells. We see various states of division and differentiation as those hematopoietic stem cells mature. So they're going to change as they grow. As we're going down our path towards our red blood cells, our erythrocytes, we're going to notice that cell is going to get smaller and it's going to start to lose a bunch of the parts that we see in our other blood cells, right? So when we start out, we can see like a nucleus and a genome and stuff in our stem cell and in these cells leading us to the erythrocytes. But by the time we get to our mature red blood cells, those erythrocytes, we're going to notice that it's missing a lot of the typical cell parts that we see in, say, our leukocytes, our white blood cells. Okay. So the plasma was just water, 90% water. It's got some proteins in there, those 6 to 8% proteins. So we were thinking about those when we thought about that osmotic or oncotic pull on fluid, right? We were thinking about the osmotic pressure of the capillary. We were really thinking about those proteins in the plasma pulling on fluid towards themselves, things that can freely move around in the plasma and therefore would get filtered along with plasma out towards the tissue would be our electrolytes, which are basically ions, right? So sodium, chloride, hydrogen, bicarbonate, potassium, calcium, those move along with the plasma. Okay. Our erythrocytes are going to be kind of stuck inside our cardiovascular system, which is good. Our red blood cells are not going to get filtered out into the tissue unless something's wrong. Okay. So that 45-ish% of your blood that's made up of erythrocytes is about 5 billion red blood cells per milliliter of blood. If we're looking at the structure of a red blood cell, we're going to see that it's a biconcave disc, right? So biconcave means concave on both sides, right? So it's kind of flat. It's got a divot on top. It's got a divot on the bottom. And what we've done by making this red blood cell biconcave like that is we've given it lots and lots and lots of surface area. We're trying to kind of maximize how much surface there is to this cell. Because when we talk about red blood cells, most of what we're going to talk about is carrying gases around, especially oxygen, right? So we want this large surface on the red blood cell so we can have oxygen diffusing in and out of the red blood cell as we load it on and off hemoglobin. So that's what's going on with the shape here. Lots of surface area so we can have lots of diffusion. So in terms of diameter, they're kind of flat, right? So they're about 8 micrometers across, about 2 thick. And I believe you did some calculations about red blood cells in this lab. It's been a while since I was in the phys lab. Okay. So what we're also going to notice about our red blood cells is that they're going to be kind of flexible. That membrane outside of the red blood cell is flexible, it's movable. If we look inside the erythrocyte, inside that red blood cell, we're going to notice a bunch of stuff missing. And so I was pointing out how those white blood cells, they've like got a nucleus. Your red blood cells don't, so they don't have a nucleus. And they also don't have organelles. So there's an impact of that fact that they don't have organelles, right? They don't have organelles, that means they don't have mitochondria, because that is an organelle, which means they're not going to be able to use the mitochondria to do like oxidative phosphorylation for ATP production, right? Because they don't have it, right? They actually also aren't particularly good at the glycolysis. So in general, they're not going to be creating a bunch of ATP in the red blood cells. They're really just going to be kind of swimming around carrying oxygen and a little bit of carbon dioxide. They're not really doing jobs. This means that they're also going to be recycled regularly, which we'll get to in a second. But let's take a quick look at how they are going to be carrying these gases that we're going to care about. Okay, so hemoglobin is a protein made up of 4 subunits. So you can see 1, 2, these ones are kind of in front. Those lighter looking ones are supposed to be in back. So 3, 4 groups to this hemoglobin molecule, right? So the globin part is this loopy part in red that's supposed to be showing you that crumpled tertiary structure of these subunits. And then the quaternary structure of the protein is all of those subunits together. This little flowery bit in the center of each of those subgroups is the heme part of the hemoglobin. Okay, so the heme part is this little yellow flower. The globin part are those red loopy bits. Okay, and the heme part is where we actually carry oxygen. So each heme group has iron in the center. So that's that Fe is the iron. Right. So that iron is what's going to allow you to carry lots of oxygen, right? You can have a little bit of oxygen dissolved in your plasma, but it's only about 1.5% of the total oxygen that's swimming around in your blood. So most of it is not just in the water in your plasma. Most of that oxygen in your body, almost 99%, is going to end up binding to iron in this heme group in the hemoglobin molecule. Right? So hemoglobin is in your red blood cells. It's in the erythrocytes. So they kind of make the hemoglobin before they lose their nucleus, their mitochondria, et cetera, et cetera. All right. That's why They lose it later rather than not having it ever. When we look at blood, part of the reason we can see a color difference has to do with that hemoglobin. So when oxygen is bound to the iron in that heme group, oxygenated hemoglobin is a brighter red than deoxygenated hemoglobin. So there's a color change. There isn't much oxygen bound to it. It's dark red. Hemoglobin does also bind to carbon dioxide, hydrogen ions, and most problematically, actually, to carbon monoxide. The hemoglobin molecule actually prefers carbon monoxide to oxygen, which is why you need carbon monoxide monitors in your apartment or in your house, right? Because if you breathe that in, your hemoglobin's going to soak that up instead of oxygen. And then your body's going to have no oxygen, brain's going to have no oxygen. Bad time. But for right now, we just want to know hemoglobin carrying oxygen bound to that heme group containing iron in the hemoglobin molecule in a red blood cell. Another piece that we're going to need to tack on when we talk about transport of gases is something called carbonic anhydrase. Okay, so carbonic anhydrase is an enzyme. And what it does is it helps us convert carbon dioxide and water eventually to hydrogen ions and bicarbonate ions, okay? So if we take carbon dioxide and water, we have a reversible reaction involving carbonic anhydrase that creates carbonic acid. And then there's another reversible reaction that can convert carbonic acid to hydrogen ions and bicarbonate ions. And all of these work in the reverse as well. So when we eventually talk about blood pH, this is why we're going to think about carbon dioxide as like a biological acid because of what happens when carbonic anhydrase causes carbon dioxide to become carbonic acid. And this reaction is also part of how we're going to be transporting carbon dioxide in the blood. Okay, so this is going to be critical to kind of like moving carbon dioxide around and to making bicarbonate ions later on as well. Right. So we'll come back to carbon dioxide later. Okay. I pulled this picture from later in your slides, but we're gonna just briefly go through this life cycle of a red blood cell, an erythrocyte. Okay. So they're going to come from those hematopoietic stem cells. They don't have any DNA, they don't have any organelles, they don't even have any RNA. No mitosis going on once you have a mature red blood cell. We're just stuck with what we got. Really bad at making ATP. So basically, all of this kind of adds up to, they're going to have a relatively short lifespan. Okay? 120 days, which may or may not seem short to you, but it is short for a lot of cells in your body. You replace about 3 million of them per second. So about 200 billion red blood cells in your body are getting replaced per day. So you're kind of constantly producing them, right? And constantly filtering them out, seeing them die off, breaking them down. Okay. So we're going to walk through all the pieces, but what we're going to see is that these red blood cells are going to be created in the red bone marrow when we get a signal from our hormones causing erythropoiesis, this production of erythrocytes. We get a signal to produce more red blood cells from the kidneys. From a hormone called erythropoietin. Your blood cells circle around, circle around, circle around for those 120 days. When it is their time to die, they get filtered out by the spleen and the liver. And the important job of the spleen and the liver, which this picture is tracing through later, is that we want to recycle that iron, right, that's in the heme groups. You don't want to have to constantly be eating more and more and more iron to maintain your iron levels. So your spleen and your liver have the important job of appropriately breaking down those red blood cells so that we can keep some of that iron to use in all those new red blood cells that we're constantly making. Right. So when we say erythrocyte synthesis, what we're talking about is making those new red blood cells. So the hormone involved that's stimulating this production is called erythropoietin, right? So we can see that referring to the erythrocytes, aka the red blood cells, poietin. I don't know, that's probably Greek. I don't know what that part means, sorry. Okay, but this is our hormone that's helping us synthesize red blood cells. So this is a signal that's coming from the kidneys. It comes from the kidneys under conditions of low oxygen levels in the blood. So your kidneys are filtering blood. Okay, so they're sensing everything that's in the blood. If that blood coming to the kidneys doesn't have much oxygen, oxygen has to be carried on the hemoglobin for the most part, right? Like almost 99% of it is on hemoglobin. So if your kidneys are noticing, hey, there's no oxygen in this blood, there's no oxygen, a good kind of assumption there would be like, okay, we need more hemoglobin to carry the oxygen, which means we probably want some more red blood cells, right? Because red blood cells are where we have that hemoglobin. So the kidneys are sensing low oxygen levels, and they stimulate the production of these red blood cells by producing this erythropoietin, which is also linked to another hormone called hematopoietic growth factor. Okay. So this erythropoietin is triggering any stem cells in that bone marrow, in that red bone marrow, to start to divide and differentiate and become mature red blood cells, mature erythrocytes. So as they're developing, so we're starting with those hematopoietic stem cells, right? As they're going on their journey towards becoming a mature red blood cells, before they lose their RNA, their mitochondria, their nucleus, they're going to make that hemoglobin, right? Because you need a gene to read how do I make the protein that we're going to call hemoglobin. You need a bunch of ATP, you need mitochondria to make that ATP. So we make the hemoglobin early on in the process. So like as that stem cell is dividing and going on its way towards becoming a red blood cell. Okay. And then just at the end of the process, to make them nice and small and agile and fitting capillaries and have a really big surface area, we eventually lose the nucleus and organelles after we already have that hemoglobin released. So we'll finish out this life cycle of a red blood cell on Friday and then do a little bit review before our exam on Monday.